I would like to present our recently result, and the title of our presentation is a cell impedance sensor chip for cancer cell detection with single cell resolution. And my name is Tien Nguyen. I come from Vietnam, and now I'm PhD student in IMTEK laboratory for sensor under the supervisor Professor Gerald University of Freiburg. And this is the outline of my talk. It consists of 5 parts. I will introduce about motivation of our work. The next part is the design of our sensor chip, and the experiment method is some result discussion, and the last part is a conclusion. So the cancer cell migration and cell migration assay. So in this figure, you can see the way of the cancer cell will metastasis in our body. So with the single cell will delaminate from the initial tumor cell and invade through the vessel and come to the other part of the body, and they will grow up and grow up. At the different state of the disease and with the different cell line, so the level of migration is different. And there have already some equipment in lab can be used for detect the migration of cell. Here's the Boyden chamber, have already used a long time. And this equipment consists of 2 apartments, the upper and below. So in the upper apartment, they will put the population of cell inside the medium. And in the below apartment, they will put some attraction. And cell will move up, move down through some hole here. And with this device will, after cell move through, and they will count how many cells can pass through the holder here. The disadvantage of this equipment is they cannot detect the signal from the initial state of cell. So after cell move through here, so they can count or somehow to detect. So, but they cannot find out some initial signal. So that's why we need to single cell analysis. So you can see here, when we put a lot of cell on top of electrodes, if we measure the average signal, the red line here, we can record only 1 signal. But the different single cell will express a different way. Also here the red line is the average signal when we measure with a lot of cells. But the different cell line is a different express the signal. So that's why we need to single cell analysis. And with single cell, the way the movement is also easier compared with the population of cell. So there is one method have already used long time to use in the cell-based sensor. It's called electric cell substrate impedance sensing, ECIS. It's invented by Gavier and Kessler. And in brief, the basic idea is concept of electrode or electrode array. And normally it's it's considered of 1 counter electrode and 1 working electrode, and they will apply the voltage here, and they will measure impedance. So the current will come from the counter to the working electrode, but when cell present on top of electrode, they will induce the different impedance. And with this method, they can real-time monitoring of cell on the substrate. But they also, by this, is when they measure, they also random seed cell on top of electrode randomly. And so that's why we need to single cell analysis. So based on that motivation, so we design our chip to measure with only 1 cell. Our chip here consists of 3 main parts. First is the microelectrode electrode array, a big electrode in the center, like a ECIS method, and 4 columns of the small electrode array symmetrically between of the middle of the big one. So depend on the direction of flow inject inside. So one side will be used for working, and the other side without cell is a reference. And the second part of our chip is a cell capture array. You can see here is one magnified figure of the cell capture. Here's one structure for trap cell. This structure consists of 2 blocks, and with a small slot in the middle of 2 blocks, so the flow still can come through when cell trap here. And when cell trap, this slot can make sure the cell stay in the cell capture structure. And the microfluidic channel, it is produced by we align a PDMS on our chip to produce the inlet, and the outlet is dished by the leader. And our chip, we integrated one method called passive pumping method. And this method is utilize the Young-Laplace equation. And when we put and when we place 2 different curvature of solution in the inlet and the outlet, so the different pressure here will induce the flow. So by utilize this method, passive pumping method, is make very, very helpful when we use with the cell-based sensor, especially in the microfluidic channel, because you only need to one time to use the pipette to fill the channel by medium, and after that you just put a drop in the one side, and we can induce the flow inside the channel. And our fabrication process consists of 4 main parts. So first, the electrode structure is defined on the Pyrex wafer by lift-off method, and after that we use the passivation to cover the channel. And next is we will produce the adhered structure to produce the cell capture structure on the microelectrodes. And the last part, we manually align the PDMS cover to produce the microchannel. And this parameter for the electrode, big electrode and small electrode, and the adhered structure, and the cell capture structure, and the microfluidic, the thickness of the microchannel is 40 micrometer. And here is the HCM image of our single cell capture structure with 2 blocks and the gold electrode here. So for the experimental protocol, we use for 2D cell culture medium system. So the chip first is cleaned by standard process, and the PDMS is covered to produce the microchannel inside. After that, we use pipette only one time to inject the fibrinolytin inside. And for example, in this step, before we cover PDMS, so the chip is immersed to some monolayer, the gold surface, to better with the fibrinolytin. So we modify the gold surface by cell assembly monolayer first, and after that inject fibrinolytin inside. And so we need to inject only one time with fibrinolytin, and after that the cell suspension in medium, we put one drop here, and the flow come in, and the single cell will be trapped here. And by this, we use for monitor the cell behavior on top of working electrodes in the medium. We call it a 2D culture system. And cell will spread, and cell with the micromotion we can detect by impedance change. And the second step is we use our chip for 3D cell culture migration assay. And so the chip in 2D after culture in incubator. So we will replace the medium inside the chamber by matrix cell. And we will incubate for 15 minutes more to cell or to matrix cell will become the polymerization. And after that, the PDMS will be lift, we will take out. And a membrane with the porous membrane here will be touched on the tape to produce the definite 100 thickness of matrix cell here. And after that, we will put a drop of medium with chemoattractant inside. And so by this step, there is a gradient will from the top to down, and when cell will be attracted by this gradient. And our expect that so cell will move up to the chemoattractant from the working electrode and will move up in the Z direction. And here I would like to present some result. So this figure is our real image of our chip, and the channel with the single cell on the electrode in the left side, and the right side, because by the way we arrange the cell capture structure is without cell we use for reference. And we would like to know how the effect of the flow rate inside the channel. We model the by using the solar zone by the console. So if in this figure it's express the flow rate distribution inside the micro channel, and you can see here this region is in the cell capture region. So the magnitude of the flow rate inside in the cell capture region is very small compared with the other, with the surrounding. So when cell will be trapped here, so cell will stay in the capture. And we use the solar zone to measure impedance with the voltage applied 10 millivolt, and for the sweep frequency we use 100 to 1 mega. And when we measure time, we measure at 4 kilohertz frequency. And the electrical equivalent circuit is built up to model the cell electrode system, and the cell is modeled by the parallel circuit consist of the capacitor and resistor, and the interface is modeled by the CPE and one charge transfer function. So this figure is the border plot of the when cell on top of the microelectrode in 2D system with the paragon electrodes and with the electrodes modified by SAM and fibrolectin. So with the modified electrodes, so cell spread attack is better, so impedance increase compared with the paragon electrodes. And by the simulate by electrical equivalent circuit, so the value of capacitor is increased about 14%, and the value of the resistor here is decreased. So it corresponds with the other literature, and here's the parameter. And this is the result when we monitor real time on the 2D system. And the insert figure, is the impedance change when we check cell. So depend on the dimension of cell, the impedance is increased about 50 kilo ohm. So when cell come to the chap structure, and all working electrode here we can check with the single cell. And here's the result when we monitor with we can distinguish the migration of two different cell lines. So here you can see the change a lot of the MDA cell because the level of migration is higher compared with the MCF7. And the blue here is the MCF7 signal is less metastatic, so the signal is not changed. Compare, it's nearly the same the reference one. So by this chip we can distinguish the migration level of two different cell lines. So here's some conclusion of our work. So our chip can be checked single cell, can real-time monitoring in 2D culture system and in the 3D culture. So we would like to thank the project and Vietnam National Education for support Thailand and the Professor Ballet for pre-sale for us. And thank you for your attention. And I would like to answer one question.